Good morning, everyone, and welcome to the TLDR, or Too Long Did Not Read presentation about our team, which officially is called Data Science Team 1 at the Translational Neuroscience Division of the Liver Institute for Brain Development. Although more colloquially, we go by the Bioconductor-powered Team Data Science group. We last presented a TLDR presentation on 2022, where we covered some of the analysis that the team has expertise on, but it's been a while since 2022, so we decided to go ahead and update everyone about what is the team currently have expertise on. And so this presentation was orchestrated by Luis, who's a staff scientist at the team, and take it away. Thank you.
Hello everyone, I am Manisha, and today I'll be presenting on specially resolved transcriptomics with Visium. To begin with, Visium technologies by 10x Genomics are the tools for specially resolved transcriptomics. It enables us to map the RNA expression onto tissue architecture with spatial precision. Here on the screen, we see the standard Visium and the Visium site assist slides. The platforms, while standard Visium is ideal for the fresh frozen tissues, Visium site assist extends the capabilities to fixed frozens and the FFPE samples. The only caveat to this is customizing the probe sets for species other than the human and mice. Next, before we get the data for analyzing, it goes through a lot of wet bench work. Here is a pipeline workflow for the Visium site assist, where the people are following the 10x pipeline and the protocols. On the wet bench side, Kristen, Stephanie, and Sarah and Ruth are the people behind the Visium site assist who also send the data for sequencing. And then Ryan is the person who runs the space ranger count pipeline, and then helps us, gives us the outputs, space ranger outputs for data analysis. To learn more about the space ranger metrics, you can take a look at our video on space, and also identifying the technical artifacts like the hangnail, the folds, the rips of the tears, or also on the spot level where there are high mitochondrial ratio, low sum of UMI, low sum of genes. The aim is to identify and remove the low quality spots, visualize and discard the outliers, and only keep the spots which are tissue covered. For this, we use scRNA, which uses the matrices, and spot sweeper, which uses the information from the neighboring spatial spots. Then moving on, we go for the feature selection once the data is cleaned to identify the most informative genes. In this, we focus on highly variable genes and spatially variable genes, to identify and understand the crucial patterns. After the feature selection, we go on to the dimensionality reduction using methods like GLM-PCA, UMAP, and t-SNE. It allows us to visualize the complex data in lower dimensions and identifying the patterns. After the dimensionality reduction, we also do the batch correction to account for the technical variations between the samples using methods like harmony, so that we ensure that our results reflect true biological differences. Then moving on, we do the clustering using the spatial algorithms, spatial clustering algorithms like Bayes-Space. Here, Bayes-Space, different resolutions of Bayes-Space clustering helps us answer different biological questions, such as at K2, Bayes-Space helps us separate the gray and the white matter, in this particular figure for spatial DLPFC. At K9 for this study, we found the best iterated histological layers. And once the clustering is done, they are annotated against the manually ground truth layers and the layers associated with histological features. Then moving on is the spatial registration step, which is the key step in our Visium data analysis. Spatial registration is an analysis that compares the gene expression of groups in query RNA-seq dataset to groups in a reference spatially resolved RNA-seq dataset. It adds the anatomical context. We map the spatial domains to the Maynard et al. Manually annotated layers. We correlate the enrichment T statistics for top markers for reference, and then highlight the most strongly associated histological layers to add the biological context. Following this, we embed our projects into the special LIBD apps. It helps us to visualize and annotate the data. We can look at the spot level data, clusters, and interactively inspect the data as well. We make it at different steps, like make after the space ranger, and also publicly share it after the publication results, and update with the results as we generate it. We have it for various projects like special LIBD, special DLPFC, Visium, SPG, AD, and many more. After this, we also document our work with packages in Bioconductor, and then also have different vignettes as a reference to understand more about these packages. In summary, we use standard Visium and Visium site assist workflows. It helps us analyze diverse tissue types to spatial transcriptomics and developmental brain disorder research.
Hi, everyone. I'm Nick Eagles, and right now I'll be presenting on an extension of Visium called Visium HD. So just a little bit of background on HD. It's quite similar to Visium standard, but actually it comes in square bins at 3 different resolutions, which we have a little diagram on the right side, the 3 different resolutions. The 8-micron bin size is what's recommended for analysis by 10x Genomics. And this gives us about 700,000 bins, which is already much larger, almost 100 times larger than Visium standard, so it's a lot of data. So you can work with that bin resolution, but you can also, at the 2-micron resolution, it's actually subcellular, so you can combine that with segmentation to form individual cells. So at different parts of this presentation, I'll be talking about bin level analysis with the 8-micron bins versus cell level, which is aggregating counts from the 2-micron bins into individual cells. So starting off, some of the goals are to just explore what software is out there. It's a very recent, Visium HD is a very recent development. So we're exploring what software tools are available, what runs on our data, and developing code when there's holes. And then at the same time, while we're exploring the software, we also want to gain interesting biological insights in the data that we're working with. And since we have spatially resolved cells, there's a lot of new questions that we can answer. Some challenges are the data is gigantic. Like I mentioned, it's about 100 times larger than that Visium standard sample, and there's not much software out there, so there's a lot to develop. So now I'll jump into some results. So some of the results are similar to the results that we're interested in with Visium standard, including finding spatially variable genes. So a trick here that we have to do is we actually counterintuitively, we have to lower the resolution with a tool called SE-raster. That paper showed that you can get similar quality results at a much faster rate if you lower the resolution. And then we compute spatially variable genes with NN-SVG. So we get some familiar genes like MBP, and we also have some genes like CALB2 and TSPAN13 that are showing sort of outlining the habenula, which is the region that we're studying in this data set. Another interesting tool is called Fixture, and this is basically taking the 2-micron bins, which are subcellular, and then doing a form of clustering. So we have a little inset here showing just the resolution that you get with this tool. And so this has been really useful for drawing out some of the biology of the tissue, such as the location of the habenula and then location of white matter tracks as well. So this is a really promising tool. Another tool, now we're talking about the cell level. So each of these spots is a cell on these plots, but this tool is called Banksy, and I've showed a few different clustering resolutions here. So we can get a lot of different we can extract a lot of biological information from these different values of K. So this is another promising tool that we'll explore a little bit more of. Another question that we're interested in answering is how are cell types distributed spatially? So we show two different approaches here. On the left, we're taking some unsupervised clusters. We're using a single nucleus reference data set, and we're asking what cell type each cluster is most similar to. On the right, we're just asking for individual cells by expression, what cell type they are based on the expression. So two different approaches to try to locate where the cell types are situated spatially. And this process involved a lot of exploration of different software tools. So we tried to internally keep track of which ones we've explored, which ones run on our data, and then different details about like how long it takes to run each tool, how much memory it takes, and useful information like that. On the top right, I've shown some screenshots of GitHub issues. So this has involved a lot of communication back and forth with the authors of different software tools for Visium HG data. So in summary, some promising software that we've already explored includes Fixture, Bin2Cell, Banksy, and SE-Raster. We're still developing novel code, which we have to be really careful with how much memory it uses based on the size of the data. And Bin2Cell in particular gives us spatially resolved cells, and there's a lot of downstream possibilities with that information that weren't possible before. And this is a project with a lot of different collaborators. So some of these are listed below.
So hello everybody, my name is Melissa Mayenquiros. I'm a remote member of the lab, and I'll be presenting today single cell single nucleus RNA-seq data analysis. So next please. I want to start reminding you that there are various single cell methods, each different on how they isolate and barcode individual cells. But in our lab, we use Translational, which is a droplet-based microfluidic method. In this, each gem, which is a gel bed-in emulsion, contains a single cell, barcode gel bed, and RT reagent. The cell is sliced, releasing the RNA, which will bind to barcode primers. And this barcode will link the cDNA from the stem cell, allowing the transcript assignment during the sequencing. Unlike bulk RNA-seq, which captures the gene expression of across all the cells in the sample, single RNA-seq will enable the analysis of individual cell profiles. So this will allow us to distinguish heterogeneous cell population and identify subpopulation that otherwise would be masked. Next, please. So in the particular project that I work on, we use avenula samples from mice to investigate the HBBTH projector neurons. The samples were generated and collected by Egege Silves under the guidance of Christine Maynard. And once the sequencing was completed, Ryan Miller processed the data using CellRanger. CellRanger is a bioinformatics suite provided by the Next Genomics, and it demultiplexes raw VCL sequencing files into FASTQ files. Then it aligns it to a reference genome and assigns them to individual cells using the unique barcodes. Finally, it will quantify the gene expression to produce the gene cell expression matrix. Next, please. Then, for the quality control, the first thing we did was remove the empty droplets, which in here only contains ambient RNA, and also to remove the doublets in case that two cells were captured in the same droplet. This was done using empty drop to distinguish the real cell and scDPL finder, a package from Double Detection. A particular feature of scDPL finder is that it simulates artificial doublets, then computate their local density to real cells, and assign a doublet score to each cell. Then we use the SCRAM package to filter out low-quality cells based on three key metrics, which are more standard in normal QC, which are the low counts, low UV counts, also low detecting genes, and finally cells with high mitochondrial RNA percentage. Next, please. Then for dimensionality reduction and batch correction, we use a GLM PCA and harmony approach respectively. And this is because PCA, traditional PCA, assumes normally distributed data, which is not ideal for sparse count matrices in single RNA-seq. So instead, GLM-PCA applies a generalized linear model that will better capture underlying structures, modeling appropriate distributions like Poisson or negative binomial. Also, before applying GLM-PCA, we perform feature selection, where we rank genes by binomial defiance. Then for, as you know, batch effects can obscure real biological reactions. So harmony, unlike other approaches, identify batch effects within a low-dimensional PCA space and correct them without altering the biological structure through an iterative process. Next, please. Then one of the most important steps in single-cell sequencing is the clustering to identify the cell population. In our case, we use a graph-based approach where the first step was to construct a SNN graph or shared nearest neighbor graph. To build this graph, we have to identify the k nearest neighbor of a cell in the harmony-corrected PCA space, then define the edges, which means to measure how many nearest neighbors they have in common, and finally to weight the edges. Once we have the SN graph, we apply the walk graph community detection, which works by simulating random walks on the graph. So the cells that frequently occur in the same neighborhood will group together. A key element of this process is the choice of k, which is the number of neighbors useful for building the graph. Because if k is too low, the graph can become too fragmented, and if it's too high, different cell types could mix. So the optimal k, of course, depends on the data complexity, and in our case, we use different numbers of k, build the graphs, and then compare their modularity scores to see which graph was the best. Next, please. Now that we identify the clusters, we can move to the downstream analysis to understand the biological significance. In our case, we identified marker genes for each cluster using the Combo-Paris functions such as 1 versus all and mean ratio. This analysis allowed us to determine genes that were uniquely or highly expressed in specific clusters, and then we create plots for the top marker genes as well as for already known in the literature marker genes. With this, we perform manual cluster annotation. For our specific research question, we also conducted differential expression analysis to compare projector neuron-containing clusters versus other neuronal clusters using a pseudo-bulk approach in which we aggregate single-cell expression at the cluster level. With this, we can use tools that traditionally are used for bulk RNA-seq analysis. And finally, we perform gene ontology and cat pathway analysis on the differentially expressed genes that we identified. This helps us to gain insight into the functional difference between these neuronal subpopulations. Next, please. So in summary, single-cell RNA-seq enables to study cellular heterogeneity to distinguish different cell types and different cell states. In our case, it helped us to characterize HVBTA projection neurons and to compare their differences between them and other brain cell populations.
Hi, this is Cynthia Soto, and I'm going to speak about the multi-ome strategy. The multi-ome strategy contains a collection of software that is used to analyze chromium single-cell multi-ome data, connecting the gene expression and the chromatin accessibility information for enhance genomic understanding, so we can, for example, study or investigate gene regulatory patterns. Broadly speaking, the multi-ome strategy starts applying the multi-ome protocol, which consists in extract nuclei from the cell and then build 2 separate libraries for each one of the modalities, one for attack and one for gene expression, and then sequencing the data and preprocessing the data. The preprocessing step is, this is a step when we basically count the number of the features that contains each one of the modalities. So it starts with raw data. And well, the recommended pipeline is the scRNA-seq, which is very convenient because in few steps we can have the count for gene expression, as well as the count from the chromatin accessibility data, and we can have an integrated data set, which is very suitable for started downstream analysis. But we are also aware that not all the data fits under the requirements to run this pipeline. So when this is the case, we can also be able to run a customized approach, which is basically in which basically we can run different pipelines separately for each one of the modalities, and then we can reintegrate the data so we can have a very similar data set to continue the downstream analysis. I want to mention in this step that there is a lot of people involved in the project. So there is, for example, Kelsey, Kristen, Ryan, Sarah, and Leonardo, staff scientists and investigators, as well as external collaborators like Lisa Johnson and Ivy Prima from the 10x Genomic Support that helps during all the analysis. From the downstream analysis, actually there are pipelines that can embed all modalities in the same data structure, and also contains a wide collection of functions, but also about also methodologies, when we can have, let's say, a very convenient workflow to work with the single-cell chromatin analysis workflow. In this example, we have several insignia that is very convenient for the multi-ome analysis with this kind of data, and also because it contains a wide collection of visualization tools when we can explore the data. And this can be applied from a regular analysis to an advanced analysis. As well, we are also aware that sometimes it's necessary to look the data with different perspectives or apply additional controls. So this slide is just to illustrate that we can analyze the data separately, applying regular tools designed for single-cell analysis. So this is one example. For example, apply it to standard quality controls for gene expression. So we can take only this modality, and for example, with regular standard analysis to detect outliers in mitochondrial levels, total UMIs, or total genes, we can apply additional controls from this data set. But we are also able to apply specific quality controls for the chromatin accessibility data. This is an example applied to the nucleosome signal, which is to observe the pattern in the chromatin structure, as well as the transcription star size enrichment score, which is a very established, well-established metric to measure the ability of the transposasa to capture the nucleosome-free regions in the chromatin. So from here, there is very similar steps that are applied for single-cell analysis. So we usually normalize, scale data, perform some batch correction, and make the clustering, which in this stage is very intensive because it's usually made separately at the initial steps. So one of the strategies is to apply the nearest network for the RNA side and the latent semantic index from the ATAC side. So this is a good, let's say, point to start a clustering evaluation and optimization, as well as to identify these cell types that are of interest for a biological question, as well as to identify regulatory elements that can be interesting for our case. After this, the real, let's say, major step in this strategy is to have the integration of this data in just one representation from the both modalities. So for do this, we have the weighted nearest network, sorry. There is a multimodal clustering that is applied in the reductions for each one of the modalities. So we have at the end cluster representing both modalities, so we can run the annotations and apply different strategies. We can use different visualization tools to look for the chromatin tracks and to identify accessible regions and boundaries over genes or the regulatory elements implicated in our genes of interest. So in summary, the multi-ome approach is a strategy to link gene expression to regulation, and it's usually used to reveal these mechanisms. And in our particular case, in our Habenula project, we explore these regulatory pathways. As well, we want to identify gene expression patterns in Habenula subdomains or subtypes. Thank you.
Hi, everybody. I'm Lucy Myers, and I'll be discussing our work in bulk RNA-seq deconvolution. As previously mentioned, we can study gene expression in the human brain at several different resolutions. I guess the highest resolution would be bulk RNA, or lowest resolution rather, would be bulk RNA-seq, where we're seeing the mixture of cell types represented in the data. And that is contrasted with more high resolution tech, including single nucleus RNA-seq, where we can profile individual cell type populations. So what is deconvolution? Deconvolution is a computational method that seeks to understand the cell type composition of that bulk RNA-seq and understand the heterogeneity within that tissue that we cannot see in the bulk RNA-seq data. So how we do that is use single cell RNA-seq cell type profiles to understand the cell type expression across these different genes, and then use that to estimate the breakdown of the cell types that make up that bulk RNA-seq sample. So again, utilizing cell type data, or single cell data to understand our bulk RNA-seq data better. And an important aspect of this process is marker gene selection. Our proposed method for this is mean ratio gene selection. We're finding highly specific cell types by comparing the mean expression of target cell types over the mean expression of the highest non-target cell types. So this helps us select highly specific genes that help us better estimate cell type proportions. To also understand the problem of deconvolution better and to select a best methods, we performed a deconvolution benchmark analysis. And how we did that is we designed a paired data set that included a bulk RNA-seq, single nucleus RNA-seq, and an RNA-scope orthogonal measurement of the cell type proportions. So we used the bulk and single nucleus RNA-seq to perform deconvolution, but then we needed a ground truth orthogonal measurement to compare from. So this is a challenge when I guess evaluating the performance of deconvolution is knowing exactly what that cell type, what the truth was. So we used imaging tech through RNA-scope IF to estimate cell type proportions. So then we used this data set and eventually compared the two to evaluate the performance of 6 deconvolution methods: DWLS, BISC, MUSIC, Phase Prism, HSP, and CyberSort X. Through our evaluation, we established that, or we saw that BISC and HSP were our top performing methods in our human brain data. And then how we can use this deconvolution estimates in real experiments, we've used them both for quality control, for example, removing samples from data sets that have high proportions of a neighboring region that might skew the results. And we can also use these estimates to control for cell types and downstream analysis, such as differential expression or EQTl, and then EQTl, we can actually test for cell type interactions. So those are some examples of how these estimates are quite useful in bulk RNA-seq analyses. So through our benchmark, we established our suggested deconvolution pipeline that we use at Liber. So we like to select single nucleus reference sets from the same brain region with multiple donors. 4+ is ideal. And we want to determine a cell type resolution that helps us answer our research question. So then number 2, we recommend selecting marker genes with our mean ratio method, which is available through the Decomvo Buddies package. And then 3, run deconvolution with either BISC or HSP, subset to that marker gene set. And then 4, check out those estimated proportions and apply it to downstream analysis as fit for one's analysis. Through this work, we have developed our package called Decomvo Buddies that has 3 main functions. One helps find marker genes with get mean ratio and implements a one versus all wrapper for scRNA find markers that is easily integratable with our mean ratio data. We also have tools for plotting gene expression quickly that either plots genes selected by the user or works well with our mean ratio output to help plot our marker genes annotated with helpful statistics. And then we can also access our data set that we designed for this purpose. So we can access that paired DLPFC RNA-scope single nucleus and bulk RNA-seq data set. So in summary, deconvolution helps predict cell type compositions in bulk RNA-seq data. Our work at Liber we've run a deconvolution benchmark to establish a best pipeline, determining HSPE and BISC as top methods, and also recommending that mean ratio method in our designed package. So this work is now in preprint. So the next I will also mention a little bit about our work in bulk RNA-seq. So bulk RNA-seq, we have a large library of bulk RNA-seq data at the Liber Institute. This makes up less of our work nowadays. However, we still have some, I guess, relevant analyses that we are exploring in this data. So first being EQTl analysis, EQTl standing for expression to quantitative trait loci. So basically this is identifying SNPs that are changing the expression of a gene in the either close or far proximity depending on the analysis. So down here we can see an example of that where across the genotype from samples in a study, we can see that there's effects, or at least is associated with differences in gene expression. So matching genes to SNPs, and we can see a difference across the 3 genotypes for this group. And then there's also different variety of types of EQTl analysis that help narrow in on the actual causal SNP that can maybe like impact the expression of a gene. So this has been relevant to a few of our bulk RNA-seq analyses, and to run this, we've been using TensorQTL.
So another type of analysis that we've done is called co-localization. The last analysis we did, we were looking at the EQTLs Luis just mentioned, as well as a reference data set called PGC3. This had a bunch of schizophrenia risk loci from a GWAS. So we were asking if these SNPs shared basically causal variants. We've been using the colloq package, and in particular the colloq.abf function. The analysis we did took one EQTL gene at a time, and we were considering all SNPs in a neighborhood, actually the same neighborhood that we used when finding EQTLs. And some results included that we found 16 genes to be co-localized. 2 of the gene SNP pairs were also significant as independent EQTLs. And finally, 123 of the co-localized SNPs were also significant for PGC3 risk. So this is a useful type of analysis when you have two data sets and you're asking if there's some shared, essentially some shared biology. So next I'll be talking about a tool called QSVAR. And before I jump into the software, I just want to give a little bit of background. So we work a lot with post-mortem human data. And with this type of data, there's an interval, an unavoidable interval when there's tissue that degrades before it's able to be frozen and preserved. And the complicated part about this is that different transcripts degrade at different rates. And ultimately, the effects of degradation can confound biological signal when you're doing differential expression analysis, and you can get false positives. So we did an experiment where we had tissue and we measured it at different degradation time points. And this was used to develop this QSVAR software. So QSVAR is an R package, and the goal is to reduce some of the effects of this degradation signal like I mentioned. So we try to summarize the signal in degradation-associated transcripts and then ultimately include those as covariates in a gene-level differential expression model. So Andrew Jaffe in 2017 already introduced this approach, but now we're using transcripts instead of the original express regions, which we think will be more accessible. And so at the bottom, we've showed that you can apply some of this code in just a few lines. And ultimately, we show that QSVAR can effectively remove degradation signal. So on the right, we have some plots where the y-axis is the t statistic from degradation, since we have different time points. And on the x-axis, we have case control signal. So what's really problematic is in that most differential expression models, we have a big correlation between these two. So there's a big confounding effect with our biology of interest. And maybe most surprising is that even if you account for RNA integrity number, you're not removing degradation. So on the bottom right corner, we have QSVAR. So once you apply this our model, you actually do remove this correlation effect. So a recent analysis that we've done with this is called, we've been trying to select the right number of transcripts used to find the degradation effect. So on the top right, the top three plots, we have replication on the y-axis, which is essentially asking how well differential expression results agree between two different data sets. And we think they'll agree better when we remove the effect of degradation. So we tried a different number of transcripts, and while the picture isn't super clear, the numbers of transcripts that work relatively best were 300 and 1,000. We're still exploring this, but this is more recent analysis that we've done to try to best select the number of transcripts. So in summary, we have a R package called QSVAR, and the goal is to reduce the effect of degradation and differential expression. It requires transcript level data instead of the expressed regions approach already introduced, and we have an upcoming manuscript for this.
I'm Gio Portea, presenting some of our work on identification, quantification, and annotation of transcripts captured by RNA sequencing in brain tissue for a project called CHESS-Brain. This project evolved from the original CHESS project from our Johns Hopkins collaborators, which used over 9,700 GTEx RNA-seq samples to propose previously unannotated RNA transcripts. That project relied on the string-type software to reconstruct transcripts from RNA-seq reads aligned to the entire human genome, which allowed the assembly of novel isoforms that were not found previously in reference annotations like GenCode or REF-seq. The CHESS-Brain project extends CHESS by cataloging brain-expressed transcripts specifically. Unlike CHESS, which used poly-A selected RNA-seq data from GTEx, CHESS-Brain leverages RNA-depleted or ribose-zero data at the Liber Institute, capturing more non-coding RNAs. To address the increase in intronic reads from nascent and incompletely processed RNAs that are prevalent in ribose-zero data, we collaborated with Johns Hopkins to develop string-type version 3, which models co-transcriptional splicing, distinguishing mature versus nascent RNAs, improving transcript assembly and quantification, and expanding the set of isoforms beyond those including in GenCode and REF-seq. As part of CHESS-Brain, we examined how poly-A versus ribose-zero library preparations impact brain transcriptomics in postmortem tissue. We analyzed 3 major DLPFC datasets, where samples were sequenced using both library preparations. Dataset A includes 170 paired samples with schizophrenia cases, enabling case control differential expression analysis. Dataset B used cell fractionation to compare nuclear versus cytoplasmic RNA capture. Dataset C, which is a DLPFC degradation experiment data, tracks RNA degradation over time, assessing how each method performs with increasingly degraded tissue, which is the same dataset used in the part of the dataset used in the QSVA study. Together, these datasets offer a comprehensive, multifaceted view of how library prep choices shape our ability to study brain transcriptomics in postmortem samples. Across this dataset, we compared multiple alignment metrics and examined how other differences are between transcriptomic landscapes captured by poly-A versus ribose-zero. On the left plot, we see overall gene expression differences between the 2 library preparations, confirming that ribose-zero captures non-polyadenylated transcripts, especially in long non-coding RNAs, while poly-A selection favors protein-coding genes. As expected, ribose-zero provides a broader RNA coverage. The plot on the right highlights transcript level resilience to degradation using string-type for transcript reconstruction. We found that ribose-zero consistently recovers better transcript structures, even from degraded samples. This holds across all genes, including protein coding ones. This complex visualization compares poly-A versus ribo0 in detecting schizophrenia-related gene expression changes before and after controlling for RNA quality using QSVA. The DG analysis was performed on case control samples from dataset A, which consists of brain-seq phase 1 poly-A and brain-seq phase 2 DLBFC ribo0. The diagonal shows the volcano plots of the 4DG analysis performed. Note how DG counts drop substantially after QSVA correction, suggesting many initial signals were driven by RNA degradation. Poly-A DEGs drop 90%, while ribo0 dropped 75%. Though ribo0 starts with lower exonic sequencing depth, explaining the initial lower DEG count. Lower triangle has correlation plots where ribo0 shows greater stability with better correlation between corrected and uncorrected analysis. Upper triangle shows concordance at the top plots, showing the cumulative agreement between poly-A and ribo0 is low, even decreases after QSVA correction, reinforcing the fact that the two library preparations capture distinct transcript populations. In summary, the chest brain work leads to a more comprehensive view of the isoform diversity in brain tissue by finding novel RNA transcripts directly expressed in RNA-seq data we analyze, augmenting reference annotation databases like GenCode and RefSeq. This and other developments in this project allow more accurate and comprehensive transcript level analysis, especially of the ribo0 RNA-seq data at the Liber Institute and other public RNA-seq data sets based on rRNA depletion. We're preparing also manuscripts comparing poly-A and ribo0 transcriptomics and presenting the SynTime3 advancements.
So we've already mentioned some open source software that our team develops, but there's actually a few more tools I want to go into. So Speakeasy and BioCMap have come out a few years ago, but essentially there are two different pipelines built with Nextflow. So Speakeasy is for processing bulk RNA-seq data, whereas BioCMap is for processing WGBS or just methylation data. The idea behind both of these pipelines is that you start with raw sequencing reads in the form of FASTQ files, and both pipelines do quantification steps, and ultimately you end up with analysis objects in R that you can do your interesting biological questions by doing downstream analyses. So we continue to maintain both of these tools, and they have websites like the example on the right. With Speakeasy, we've done some bug fixes, and we've also allowed quantification of transcripts for rat. Those are some updates in the past few years. So another tool is called Visium Stitched. This is an R package for integrating multiple Visium samples together. So if you have a brain region that's too large to fit into one capture area, you can use this tool to stitch multiple capture areas together and analyze the brain region in one piece. So we've done this in the nucleus accumbens in the hippocampus. We have a manuscript out for this software, and one of the key findings with the software is that if you stitch together data, you get higher quality clustering results. So you can see some of that in this plot right here. And on the right, we have an example of spatially continuous clusters that are possible once you stitch together multiple samples with Visium Stitched. Another tool we've developed is called Slurm Jobs. The main idea is that it's an R package for working with Slurm, a type of high performance computing environment from R. So one of the things it does is it simplifies repetitive tasks like creating jobs, like shell scripts for submitting R jobs. It also enables development of parallel workflows with the function called job loop. So you can efficiently process data that way. It also has functions for monitoring memory usage and other things. So for example, we have a snippet at the bottom of this job report output showing how much memory your job used, how long it took, and whether it successfully finished, among other things. So I just mentioned I went to depth on a few of the tools, but we also developed tools like spatial LIBD, and we've already mentioned Decombo Buddies, QSVAR. So we maintain quite a few different tools here. Next, I'll be talking about some of the work on the cluster that we work on called JHPCE. So one of the main things we do is we maintain what are called modules. So these are units of software that we maintain for our team and then our collaborators as well. The idea is that you can run a simple commands like the one at the bottom, module load bin to cell, for the software called bin to cell. The idea is that you install the software once for many users, and then they can use this one command to load it, and it should behave similarly for everyone, or actually the same way for everybody. And that way you get reproducible and straightforward data analysis. So on the right, we have some examples of the modules that we've developed in the past 6 months. So we've been pretty active doing this. So we also have a RStats club and a journal club that we do, and we also post the videos, which I'll get more into. So our RStats club is every Friday, and somebody typically presents a topic that involves something like genomic software, computational methods, new R packages, or tips for just working at as data scientists on the cluster. So the idea is that we're sharing knowledge and we're building skills that we might not necessarily need to discuss with the projects that we're working on, but we are constantly learning and sharing that knowledge. So on the bottom, we have a snippet of the Google Sheet that we maintain, which has YouTube links, and so that you can actually go through and search different topics. So similarly, we have a journal club, and the idea is that we actually have these YouTube playlists that are public, and so you can just search by keyword, and then this complements our efforts to share knowledge through data science guided sessions. And so this is an effective way to share what we learn with everybody.
Well, thank you very much, everyone in the team. So the current team is composed by 2 staff scientists, Cynthia and Luis, as well as a research associate, who is Nick, and then a research assistant, Manisha. I noticed that Nick's, oh no, sorry, the title is complete. You do have like all the positions that Nick has had over the years. Then we also have a few remote members who are either current students or recent graduate students from the undergraduate program in genomic sciences in Mexico, where I also studied. That is Diana Gonzalez Padilla and Melissa Mayen. But like the team has changed over time, so you want to connect with and follow what like people from the team are sharing. There's a few Blue Sky starter packs that you might be interested in. You haven't heard about Blue Sky. It's kind of like an open source replacement of Twitter or X. And the starter packs are a quick way where you can find a list of people that you might want to follow or learn more about. So that includes a starter pack for members of the team and alumni of the Data Science Team 1. And that includes Andrew Jaffe and whoever was part of the team when Andrew Jaffe was leading the team before 2020. So with that, we hope you enjoyed this presentation, and we'll see you around. Thank you.